Hello viewers, today we are going to learn about structure of nephron. Let us recapitulate the previous topics. In last class we completed about organization of human excretory system and then internal structure of kidney. Yes, today we are going to discuss about structure of the nephron. Students, what are nephron? The nephrons are nothing but the structural and functional units of kidney. So you observe the visual representation of nephron. It is nephron. Why they consider I structural unit? Let us see. Each kidney contains approximately 1 million of nephrons. So the kidney formed by nephrons. That's why the the nephron considered as what structural unit of kidney. You observe the longitudinal section of kidney in which the conical shaped structures are called renal pyramids, in which the fine hair size thin tubular structures are present. They called nephrons. Followed, the nephrons are structural units of kidney. Followed, right? Next, why the nephrons considered as functional units? Because they filters the blood and prepares the urine. They separates excretory waste material from the blood. So the entire filtering process to be takes place in the nephron. That's why the nephrons are considered as what functional units of kidney. Alright. So you observe this visual representation. In what way the blood going to enter into the nephron and going to filter and separation of the urine. So that is entire phenomena is what? Filtration phenomena. It is mediated by the nephron. That's why the nephrons consider as functional units. Right. Next, if we observe the structure of nephron, it includes various kinds of parts. They are Malpighian Corpuscle. The Malpighian Corpuscle include the Bowmans Capsule and Glomerulus. And next, after that, Uriniferous tubule. It is a long coiled tubular structure. Along its length, various kinds of parts associated. They are sequentially the proximal convoluted tubule, then loop of Henley and distal convoluted tubule, initial collecting duct and collecting duct. Okay, right. Next, besides that, some other associated structures also found along with tubular nephron. So they are as follows. You observe, they are peritubular capillary network which supplies blood to the nephron, and next Vasa Recta, and besides that Juxtra glomerular apparatus. These are the three associated structures that found with the nephron. All these components are present in nephron. Let us see the features of all these components. So if we observe all these if we observe the visual representation of nephron, let us verify all these components in the visual representation. The first, Malpighian corpuscle. You observe this is nephron diagram. So that is Bowmans capsule and then glomerulus. So the Malpighian corpuscle include two components. They are Bowmans capsule and glomerulus. You have to identify these two components in the diagram, right? Next, second, uriniferous tubule. It is a long coiled tubular structure. You verify the uriniferous tubule in the diagram. So this is uriniferous tubule, long coiled tubular structure. It includes various kinds of parts. They are sequentially. First one, proximal convoluted tubule. You verify it in diagram, that is proximal convoluted tubule. And second one, loop of Henley. You trace out, this is loop of Henley. And next, distal convoluted tubule. You verify it, distal convoluted tubule, and then initial collecting duct, and ultimately it leads into collecting duct. This is collecting duct. So we verified all the parts concerned to uriniferous tubule, and besides that some associated structures also present in nephron. The associated structures are the blood supply components, blood capillary network, and juxta glomerular apparatus. The blood capillary network differentiated as peritubular capillary network and Vasa Recta. Let us see these components in the diagram. This is structure of nephron. You verify blood capillary network present on the surface of nephron. So among them the first one peritubular capillary network, it is peritubular capillary network, followed the second one Vasa recta. So this is Vasa recta, followed. So with this we verified all the components related to nephron in the diagram. Let us discuss in detail about each and every component of nephron. So the first component of nephron, Malpighian corpuscle, it also called as the renal corpuscle. Student, remember the further names are very important. The Malpighian corpuscle also called renal corpuscle. It contains Bowmans capsule and glomerulus. You verify the diagram. This is the renal corpuscle diagram. It includes two components. The first one is Bowmans capsule and second one glomerulus. This is glomerulus. Followed? So, right. First about Bowman's capsule. What is Bowman's capsule? The scientist Bowmans revealed it. That's why it named as Bowman's capsule. It is a double layered cup-like structure. You verify in the diagram. So it is a double layered cup-like structure produced by invagination of membrane. The Bowman's capsule's outer layer is called parietal layer. You verify the visual representation. The outer layer is called parietal layer. Already I told it is a double layered cup-like structure. The wall of Bowman's capsule include two layers. The outer layer is called parietal layer. The inner layer is called visceral layer. You observe the visceral layer is produced by invagination. Followed? So due to the invagination, the visceral layer is established. Okay, clear? Right, the Bowman's capsule. The parietal layer of the Bowman's capsule formed by squamous epithelium. So it is lined with squamous epithelium. Very important. Right, next the visceral layer includes special kind of cells they called podocytes. You observe these are podocytes. Right? They are also called food cells. Right. The Bowman's capsule inner visceral layer surrounds the glomerulus. The internal part is glomerulus. So the visceral layer of Bowman's capsule completely envelops the glomerulus. So it surrounds the glomerulus. Followed? Right. So this is glomerulus. Right. Next about glomerulus. What is glomerulus? It is a tuft or bunch of capillaries. Followed? It is a bunch of capillaries. So you verify this is glomerulus. It consists complex anastomosing plexus of vessels. So it never contain isolated loops. It consists complex anastomosing plexus of vessels. Right? The glomerulus formed by afferent and efferent renal arterioles. So it formed by two arterioles. You verify this is afferent renal arteriole. It is a branch of renal artery. So the afferent renal artery supplies oxygenated blood to the kidney. So the afferent renal arteriole brings oxygenated blood into glomerulus. Next, such blood carry away by one more blood vessel, that is efferent renal arteriole. Students, remember, so the blood vessel that entering into the Bowman's capsule is afferent renal arteriole, that emerging out, that coming out from the Bowmans capsule is efferent renal arteriole. So the efferent renal arteriole supplies blood. The efferent renal arteriole collects blood from the Bowmans capsule, otherwise glomerulus. If we observe the size of the afferent and efferent renal arteriole, you verify the visual representation. The afferent renal arteriole diameter is greater than efferent renal arteriole. So, the size of afferent renal arteriole greater and size of efferent renal arteriole is lesser. That's why the pressure exerted in the glomerulus, right? Let us see the structure of glomerular membrane, otherwise filtering membrane. It is also called filtering membrane, that is produced by the fusion of wall of glomerulus and wall of Bowmans capsule. You observe this is glomerular membrane. You verify in the diagram, right? How it is produced, let us see. It is formed by two structures. They are wall of Bowman's capsule, and that is wall of Bowman's capsule, and second one is endothelial lining of glomerulus. You verify this is endothelial lining of glomerulus. Followed. Right. If we observe the filtering membrane, it includes three layers. They are sequentially from outside to inside: wall of Bowman's capsule, basement membrane, and endothelial lining of glomerulus. Right. So you verified all these three layers. This is Bowman's capsule and filtering membrane. So this is wall of Bowman's capsule and wall of glomerulus inside. In between these two layers are joined by basement membrane, so that include collagen and connective tissue. So the filtering membrane includes three layers. They are wall of Bowman's capsule and basement membrane and wall of glomerulus, right? Next, if we observe the filtering membrane, some structures to be found along the filtering membrane. They are the minute perforations to be found along the endothelium of glomerular wall. So such perforations, such apertures called fenestrae. Let us see the fenestrae on endothelium. Between the endothelial cells, minute pores are there. They called fenestrae. They useful for filtration of the blood. Right. Next, you observe. Next, besides that, if we observe the wall of Bowman's capsule, in the wall of Bowman's capsule, specialized cells are there. They called podocytes. In between the podocytes, the space to be remained, the space present between podocytes of wall of Bowman's capsule called slit pores, otherwise filtration slits. Right? You observe the foot-like cells are called podocytes, otherwise pedicels, foot cells. In between the cells space is there, so that is called slit pores. So all these slit pores nothing but filtration slits. So all these fenestrations and as well as slit pores facilitate glomerular filtration. You observe this is glomerular filtration process, right? Right. Next, if we observe structure of nephron, the subsequent part is uriniferous tubule. The uriniferous tubule is a long coiled tubular structure. It is also called a renal tubule, and it consists of various parts along its length. They perform different function. The first one is proximal convoluted tubule and loop of Henley, distal convoluted tubule, initial collecting ducts and collecting ducts. So this is organization of uriniferous tubule or renal tubule. Followed, right? Next, the first part of the uriniferous tubule, proximal convoluted tubule. If we observe the proximal the allocation of the parts of uriniferous tubule, The proximal convoluted tubule and distal convoluted tubule remained in the renal cortex portion. Already we learned section of the kidney. The internal section of kidney include cortex, outer cortex portion and inner medulla portion. So the proximal convoluted tubule and distal convoluted tubule of the nephrons lies in the cortex portion. You verify this diagram. So this is nephron and it is differentiation. The outer part is cortex and inner portion is renal medulla. You verify the PCT proximal convoluted tubule that located lies in the cortex portion, and next DCT that are present in the cortex portion. So the Malpighian Corpuscle, the PCT and DCT are located in the cortex portion. Followed? Right. What parts to be found in medulla portion? The loop of Henley and collecting ducts lies in renal medulla. You verify the visual representation. This is the medulla portion. In the medulla portion we found loop of Henley regions and as well as collecting ducts. Followed? Right. First, we will discuss about proximal convoluted tubule. The Bowmans capsule leads into proximal convoluted tubule. Its abbreviated form PCT. It is a short coiled tubular structure, lies in the cortex region. The proximal convoluted tubule lined with cuboidal epithelium. You verify the lumen of the PCT and the cells are cuboidal epithelial cells. The cuboidal epithelial cells are with a rich brush border surface, so contain many microvilli and as well as they contain more number of mitochondria. You verify the cell. These are microvilli for absorption and next mitochondria, more number of mitochondria. So metabolic rate is high of these cells. So these are the cells of PCT, right? Next, the PCT leads into loop of Henley. Let us see the features of loop of Henley. The scientist Henley revealed this part, hence it is termed as loop. So it is appear like a U-shaped tube. You observe this is loop of Henley. It is a hairpin shape, otherwise U-shaped tube that retain in medulla portion. The loop of Henley differentiated as again two regions. They are descending limb. You observe descending down. So that extended from cortex to medulla portion. You observe this is descending limb that going to extend from cortex to medulla, and next ascending limb is going to extend from medulla region to cortex region. Follow? So the Henley's loop again include descending limb and ascending limb. Follow? Right. Next, if we observe the ascending limb, it again differentiated as the proximal portion as thin segment and distal portion as thick segment. So you observe this is lower portion, it is thin segment, and next it is upper portion, it is thick segment. So the ascending limb again differentiated as thin segment and thick segment. Followed? Right. Next, if we observe the histology of the loop of Henley, the wall of loop of Henley also lined by cuboidal epithelium, but the epithelial cells are with less brush border surface and mitochondria. You verify the cells of loop of Henley, they are completely flat. Of course, they are cuboidal cells but flat. The microvilli are very less and mitochondria also less in number. So the subsequent part, distal convoluted tubule, the ascending limb of loop of Henley leads into distal convoluted tubule. You verify this is distal convoluted tubule. It is a highly coiled tubular structure that retained in cortex portion, right? If we observe histology of distal convoluted tubule, it also lined by cuboidal epithelium with irregular spaced microvilli and less mitochondria. You verify the representation, cell representation, you compare the cells, the microvilli are very less and irregularly spaced, and next mitochondria number also less in number, right? Let us see the subsequent part, collecting duct. So all the distal convoluted tubules of different nephrons opens first in initial collecting duct. The group of initial collecting ducts ultimately forms collecting ducts. So you verify this is collecting duct. Alright. If we observe the histology of collecting duct, it is also lined by cuboidal epithelium. Some portions are lined by columnar epithelium with irregular spaced microvilli and less mitochondria. You verify the visual representation. Right, duct of Bellini. All the group of collecting ducts of various nephrons directly open into a wider tube that is called duct of Bellini. It leads into calyces of renal pyramids. You observe. So this is collecting duct, either side to collecting duct. Various group of collecting ducts are there. They lead into wider tube that is called duct of Bellini, leads into calyces. Some associated structures also present in the nephron. So the associated structures are blood capillary network and Juxtra glomerular apparatus. The blood capillary network includes peritubular capillary network and Vasa Recta. Alright, let us see all these sections in detail. The efferent renal arteriole forms peritubular capillary network on PCT and as well as on DCT. Already we discussed the blood supply to the nephron. The afferent renal arteriole supplies oxygenated blood. Then it collected by what? Efferent renal arteriole. So the efferent renal arteriole comes out from the Bowmans capsule. This is efferent renal arteriole. You verify the visual representation, and it comes out from the Bowmans capsule and going to form a network on surface of the PCT and DCT. So this capillary network is called peritubular capillary network. Followed? Then one more network also found on loop of Henley, that is Vasa Recta. The same vessel, the same efferent renal arteriole going to form the same network on surface of loop of Henley. The peritubular capillary network going to form one more capillary network on the surface of loop of Henley. So that is called Vasa Recta. Followed students, this is very important. So all these minute capillaries going to fuse and ultimately forms a renal venule. You verify that you have to focus on Vasa Recta. All the capillaries that present on Vasa Recta going to merge with each other and ultimately forming a major vessel that is called renal venule. So all the renal venules of different nephrons adjoin with each other and ultimately forms the renal vein that comes out from the kidney. So the renal vein collects deoxygenated blood from the kidney. So one more structure that associated with nephron, juxtaglomerular apparatus. It is very very important. JGA. The juxtaglomerular apparatus is a sensory endocrine organ. Very very important. So it is a sensory endocrine organ that found in juxtamedullary nephrons. So juxtamedullary nephrons are one kind of nephrons. So these nephrons only contain this structure, juxtaglomerular apparatus. You verify this is juxtaglomerular apparatus, right? So in what way it established, let us see. The juxtaglomerular apparatus is a complex organ that formed by the fusion of cells of afferent or efferent renal arteriole and distal convoluted tubule. So students, you remember, so two uh, kinds of different cells. The cells concerned to blood vessel and the cells concerned to DCT, and these two cells are going to fuse, merge, and ultimately going to form juxtaglomerular apparatus. You observe the cells of afferent renal arteriole, it is a blood vessel, and cells of efferent renal arteriole, and they are going to merge with, join with cells of distal convoluted tubule, and ultimately going to form juxtaglomerular apparatus. So in the juxtaglomerular apparatus, the involving structures are afferent renal arteriole, efferent renal arteriole, and as well as DCT. Follow? So right. Next, the juxtaglomerular apparatus, the wall of DCT and wall of afferent efferent renal arteriole are joined and to form juxtaglomerular apparatus. Let us see visual representation. You observe this visual representation. So this is juxtaglomerular apparatus, the green color portion. You observe this is juxtaglomerular apparatus. Follow it. Next, this is DCT, distal convoluted tubule, and this is afferent renal arteriole, and these are efferent renal arteriole. So these three structures going to adjoin, their cells going to join with each other and form what? Juxtaglomerular apparatus. Follow students. So that is juxtaglomerular apparatus. It is a sensory endocrine organ. Let us see. The juxtaglomerular apparatus include three kinds of cells. So they are juxtaglomerular cells, they are nothing but cells of afferent renal arteriole and efferent renal arteriole, and macula densa cells, cells of DCT and Lacey cells. Right? So the juxtaglomerular apparatus constructed with the three kinds of cells. They are namely juxtaglomerular cells, macula densa cells, and Lacey cell. Juxtaglomerular cells are concerned to afferent renal arteriole and efferent renal arteriole. The macula densa cells are concerned to the wall of distal convoluted tubule, and Lacey cells are specialized cells. So the juxtaglomerular apparatus include various kinds of cells. You will see the modified smooth muscle cells of afferent renal arteriole and efferent renal arteriole called juxtaglomerular cells. You verify. So this is afferent renal arteriole and another one efferent renal arteriole. The wall of efferent renal arteriole contain endothelium lining, inner lining. The cells of endothelium undergoes modification, and such modified cells are called juxtaglomerular cells. So the modified smooth muscle cells, remember these are smooth muscle cells, the modified smooth muscle cells of afferent and efferent renal arteriole called juxtaglomerular cells. They are endocrine in function. They secrete renin hormone. The function of renin hormone learn in the regulation of the kidney. Right, next, the second variant cells, macula densa cell. The modified epithelial cells of distal convoluted tubule called macula densa. You verify this is DCT. And pink color cells along the wall of the DCT, pink color cells are there. They are modified pillar-like cells. They called macula densa. The macula densa cells are chemoreceptors. So that's why the juxtaglomerular apparatus termed as sensory and endocrine organ. Juxtaglomerular cells serves endocrine function, and macula densa cells perceives the senses signals. So hence they are chemoreceptors, so sensory organs, right? Next, let us see one more topic, that is types of nephrons. Based on allocation of nephrons, the nephrons are categorized two kinds. They are cortical nephrons. These are about 85% present. Each kidney contains approximately 1 million of nephrons, 10 lakhs of nephrons. Among the 10 lakhs of nephrons, 85% nephrons are cortical nephrons. Remember, and one more, juxtamedullary nephrons. They contributes 15%. Among the 1 million, 15% nephrons are juxtamedullary nephrons. Alright? So the nephrons are categorized two kinds according to their allocation. They are cortical nephrons. These are more in number, contributes 85%, and second one juxtamedullary nephrons about 15%. If we observe their organization, their structure, they show some differences. Allocation is different and their structure also little bit variation. So first about cortical nephrons, these are located in cortex portion of kidney. The loop of Henley of cortical nephrons too short and lies some portion in the medulla region and some portion in the cortex region. That is different. And Vasa Recta is completely absent in the cortical nephrons. Very, very important, ask an entrance examination. So the cortical nephrons are retained in the portion of renal cortex, and their loop of Henley is shorter and little portion present in medulla region, and Vasa recta is absent. You verify this cortical nephrons in the visual representation. So this is outer cortex region and inner medulla region. So these are cortical nephrons and their loop of Henley too short and a little portion present in medulla and little portion in cortex. Followed. Right. Next, Vasa recta is absent. Second variant, juxtamedullary nephrons. The juxtamedullary nephrons are larger size nephrons, and loop of Henley is longer in these nephrons, and the loop of Henley of these nephrons completely lies in the renal medulla, and prominent Vasa recta is present on surface of loop of Henley. Let us see juxtamedullary nephrons. So the outer portion is cortex, the inner portion is medulla. So these are juxtamedullary nephrons. Alright, you verify their loop of Henley. It is very long comparatively with cortical nephrons, and completely the loop of Henley arranged in medulla region and with Vasa recta. So these are the differences between cortical and juxtamedullary nephrons. You have to trace out the differences between cortical and juxtamedullary nephrons. Ask an entrance examination. Next session we are going to discuss about urine formation, otherwise physiology of nephron. Thank you.